Professor Kubo, who is the second vice president of IUPS. We have 30 minutes.
First of all, I'd like to say congratulations on the beautiful start of the ENSPS conference. So I feel really touched when I think about. And our society also organized a congress in 2019 in Kobe. So this is the content that I'm going to present today. And I really want to first to present, but finally I decided to give a sort of example lesson for student classes. Please understand as such. So it focuses on basics of ion channel study. And the second part, I'd like to mention perspective of ion channel research with very brief introduction of our research. What is this? Over or egg you have this morning? And my answer is it is cell. And then which part of a cell is most important? Maybe you say nucleus, where genetic information is stored, or mitochondria, which is important for energy production. But my answer is no. What's most important in cell is membrane. So it is order between self and non-self. Without membrane, there is no cell. So let's say this is live organism. If this is single cell organism, this is one life. And there is outside environment or extracellular environment. What's life? What are important features of life? So please understand, I'm talking this as an example lesson of physiology. Okay? Homeostasis. This is very important. And adaptation to environment. This is also indispensable for life. And of course, maintenance of species. This is also very important. So, and to achieve these features, we have to do metabolism for homeostasis, and we need to discard what's not needed, and we have to take up nutrition. So for metabolism, and for the adaptation to environment, we have to, for example, flee from enemy action, and also we need to sense environment. So input and output. These are fundamentally important for life. And for these fundamentally important life, we have membrane proteins, such as ion channels and receptors. So ion channel is very important for excitability of the membrane, and this is indispensable for activity, locomotion, and also for sensing. Receptor is important. And for the disposal or uptake of nutrition, transporters are important. So I think you understand how important membrane is and how important membrane proteins are. So do you agree with me?
Yes.
Oh, thank you. So that is the reason why we are working on ion channels. So next, I'd like to explain contribution of ion channels in membrane excitability. It is very well known Hodgkin-Huxley theory, and they recorded action potential from squid giant action. So action potential is something mysterious. From the resting potential, depolarization occurs, overshoot, and repolarize, and there is after hyperpolarization. And they tackled this mysterious action potential to know the mechanism of the action potential. And they did voltage clamp analysis to record membrane current and separated sodium-potassium conductance. And by mathematical equation, they found this equation fit the recorded current very well. So there are 6 voltage-dependent factors. And then they calculated 6 voltage-dependent factors at each membrane potential. So by getting these parameters, they got everything, and now they try to reconstitute the action potential in computer. So they successfully reconstituted the action potential. So by assuming sodium conductance and potassium conductance, which depends on membrane and the time. So and they did this work in 1950s and awarded the Nobel Prize Physiology of Medicine in 1963. So, but their achievement is truly great because they did it in 1950. They built, constructed their theory. So at that time, ion channel was no more than a notional hypothetical molecule to explain membrane excitability. So without knowing it, they built a very beautiful theory. So that's very important point, I think. This is, people now believe the presence of ion channels without doubt. And one more important thing is structure or image. And this is a very early stage image in 1980s by Nigel Alphin in UK. And this is nicotinic acetylcholine receptor. So also this is low resolution image. You can see ion permeating pathway in the center. So this is a proof of membrane protein with an ion permeation pathway. Nowadays we have much better high resolution images like this. So next, I'd like to explain about in vitro expression system for ion channel study. Why? After the isolation of cDNA gene, we can now introduce mutation to the wild type. So then we can compare wild type channel and the point mutated ion channel. So and by comparing the phenotype, we can tell, oh, this part of the protein is important for this function. So that is what we call structure-function relationship. And for that purpose, in vitro expression system is very important. Because, for example, if we record from neuronal cells, there are more than 20 types of ion channels expressed. So then we cannot tell which part of the current is from which ion channel. So that's why to know explicitly, it is necessary to express only the focused ion channel. So for that purpose, to have a cell with no endogenous ion channel current is very important. So that's the advantage of in vitro expression system experiment. And for that purpose, we use water frog. And in Japan, it is called African crow frog. So, and this is a mother, and by surgical operation, we isolate all sites, and the diameter is about 1 millimeter, and we inject the cRNA of wild type channel or mutant channel, and then compare the phenotype by 2 electron voltage clamp experiment. So this is very powerful to approach the structure-function relationship. And I'd like to now show 2 successful examples of structure-function study. One is selective permeation, and another voltage-dependent gating. So you know, sodium channel permeates only sodium, not potassium. And the potassium channel permeates potassium, not sodium. But you know, potassium ion is larger than sodium. Why bigger bigger potassium ion permeates better than small sodium ion. So it is a question of selective information. Potassium ion is larger than sodium. However, potassium channel permeates large potassium ion 1,000 times better than small ones. So what's the mechanism? And the answer was obtained by mutagenesis and also by structure analysis by Rob McKinnon, who was awarded a Nobel Prize in chemistry. And in the center of the tetrameric molecule, there is formation pathway, and this region is expanded, enlarged here. So the carbonyl oxygen binds to permeating potassium ions. And this is side view. Then let's see top view. So when potassium ion is in potassium channel, it is like this. So you can see carbonyl oxygen of the backbone touches potassium ion. However, when small sodium ion is in the pore, so it is not fully touched. So this difference is important. This is close to hydrated state of ion in water, in solution. In solution, ions are hydrated, and but to enter the pore, water is removed. However, these mimic the water molecule. So that's why energetically this is close to the hydrated state. In contrast, in the case of sodium, it is dehydrated state, which is energetically very unfavorable. So this is the mechanism of ion selectivity. And the next, why voltage-gated channels open upon depolarization? How do voltage-gated channels sense membrane potential? So it is also a fundamental question, and the answer was obtained by CD necron immediately after CD necron, because there is a very special transmembrane region which is rich in positively charged amino acid residues. So then we can easily imagine. So this segment is in the down state, the resting state, because this side is negatively charged. But upon depolarization, inside positive. So then the positively charged segment will be repelled and move upward. So people believe this is a mechanism of the voltage-dependent gating. But this is conceptually structure analysis of ion channels very much delayed. But after this, this is in a sense structure analysis without crystal. Just take purified proteins imaged by Tri-EM, and maybe there are so many bottom view photos, and make average, and averaging. By averaging, noise decreases and the signal increases. Now we can have beautiful bottom view. Also, we have top view, oblique view, side view, and combining all these refined images in computer, we can have 3D structure. So this is structure analysis without crystal. So it induced remarkable advancement of ion channel structure study. And more recently, there is another powerful approach, AlphaFold in 2021 and AlphaFold 2 in the next year. So this is computational prediction of the protein structure. So at the beginning, we did not trust it too much, but it is really powerful. So when the structure is totally unknown, the prediction is very powerful. It gives very important information. Next, about conformational changes. Structure biology is important, as I said, but it provides us with only snapshot, still photo. But we actually want to see a dynamic image of the functioning molecule. So for that purpose, of course, it is not possible to take the photo movie, but instead, people approach dynamic aspects by using various spectroscopic analysis optophysiologically. And one method is voltage grammetry, And we can record electrophysiologically the ionic current and also track the structure rearrangement by putting flow over here. So the fluorescence intensity changes depending on the change of the environment. So for example, here it is in lipid membrane and it is in water. So by recording the fluorescent intensity change, we
difference for flat solution in ST1. So ST1 is involved, calcium is involved in the regulation of fat intake. So in order to show that there is a message from control ray, we apply some chewing gum onto the tank and we observe that